I am Iván Nombela. I am a postdoctoral researcher working in Charité Universitätsmedizin Berlin. And today I will show you, I will share with you part of the research that I have been doing during the last 2 years. So first, I would like to point why it's important to study bats, right? And the reason is because the World Health Organization printed these blueprint printed guidelines where they describe which are the most concerned pathogens for humans, right? And from the list of viral pathogens, you can see that there are listed 8 of them. So 4 of them, they were discovered to have a bat origin, right? So, for example, the Nipah virus was found to be a reservoir in black flying frogs. Ebola is not coming originally from monkeys, but these monkeys probably got infected by some fruit bats in Africa. And this is the topic of the presentation. The SARS-like coronaviruses, or coronaviruses in general, they are found in these horseshoe bats that I will extend right now. So COVID-19 was a pandemic produced by SARS-CoV-2, which is a coronavirus, but in early 2003, there were some coronavirus outbreaks in China. And after 2 years of this pandemic, there were like several studies that they discovered that these Rhinolophus species bats, they were the original reservoirs of coronavirus. And you can see how these bats, they have a very characteristic nose. It's like a horseshoe shaped, and they distribute across the Mediterranean, through the Middle East, north of India, and finally China. So now I would like to get in deep to explain you why bats are so special. I mean, as you know, probably bats are mammals, and it's the only mammal with ability to fly. But flying implies like many things from the bat. They need to consume a lot of energy, and this flying activity is producing like a fever-like syndrome in the bat, right? So imagine if you have like fever every day. I mean, you will be under a very like a stress condition, not like emotional condition, but also like physical stress, right? So during the evolution, which has taken like millions of years in bats and also in other species, of course, the bats develop like a better control of the inflammation produced by this fever. So that's the main theory that points towards like why bats they have a better control of this inflammation and diseases, especially viral diseases. So with all this information in mind, we asked ourselves the research question: why bats can harbor coronaviruses without showing any sign of disease or sickness? So before we are working with coronaviruses, so we are going to head to the research laboratory. This is like a high biosafety lab, and that's a picture of me, and that's like the type of equipment that we need to wear. It's like a complete isolation from the environment, so we are not exposed to the coronavirus by any means. And here you can see that I have a helmet with assisted ventilation that you cannot see, but the ventilator is connected to a tube to the helmet. I'm wearing also like double gloves and a waterproof lab coat and boots, just to guarantee this total isolation from the environment. So we went to the lab and we infected like human cells and bat cells. This is like a comparative point of view. This is like when you look at the microscope at the human cells, that's how it looks on the left. You can see that most cells are rounded and they are very bright. All these are dead cells. When you look at the bat cells, you can see that some cells are dead. Yeah, you can see that there are these rounded bright cells, but you can also see that there are like many stretched cells attached to the plate, right? So that means that these are like live cells. So it's interesting to see that these lung cells from bats they can resist the SARS-CoV-2 infection. In the laboratory, in this biosafety 3 laboratory, we can do many things apart from infecting, but we can also manipulate the coronaviruses. We can edit the genome and include many modifications, right? You have to go and understand the genome as the molecule that contains all the information about how you are. All the living organisms, they have a genome. So in the species laboratory, we can take the viral genome of the coronavirus and we can remove genes from the genome. We can also introduce specific mutations, or we can even include genes. And that's like very interesting because we did the following. As you can see in this slide, we introduce a protein in the SARS-CoV-2 genome, which is fluorescent. It's like a protein from these jellyfish. Probably they use this like protein to communicate between each other, but it's like fluorescent protein, right? So what you will expect to introduce this protein in the SARS-CoV-2 is that when SARS-CoV-2 is infecting a cell, this cell is going to become fluorescent, and you will see how the cells are turning green. And now I will show you how this replication is happening in bat and human cells through an analysis of this green fluorescent. So I will show you two videos. In magenta, you can see where are the cells located. Here, for example, there is one cell that is found here. This one is infected. And in green, you can find the virus, right? So I am playing the video, and you will see how this cell is turning green. You see how the virus is replicating more and more and more. And after like 20 hours, the cell completely dies. Yeah. And you see that the virus is released. If you are interested, I will just replay the video again. So that's what we observe in the case of humans, right? You see that the cell survives 20 hours, and there is like viral replication going. But this is what happened in bats. This is a bat cell. You see how they start becoming like green. The cell survives with a huge amount of virus inside for, again, more or less 18 hours. And then the cell completely dies, right? I don't know if you notice like a detail. Okay, I will go to the next slide. This is like the point of the green fluorescent, like the maximum expression of this, like green fluorescent expression in the cell, right? So as you can see how the bat cell is like very green. It's like so green, it's like becoming even white at some point. So the conclusion from this is that the bat cell can harbor more viruses, can tolerate more amount of virus inside. That's like one of the main points of the research. And then moving into the next point is like when you just analyze the whole scenario, not like a single cell, we found this. Here, it's a combination of the light microscope with the fluorescent of this green
2 minutes,
fluorescent.
Iván.
Yeah, thank you. You will see here in black and white, in the gray scale, you can see where are the cells. You can see that they're distributed across this cell monolayer. And you see how many cells are infected in the case of humans. But in the case of bats, in the monolayer, you can only find like a few cells, right? So it seems that bat cells, they can harbor more virus, but they're getting less cells infected, while the opposite is happening in humans, right? So that's the final message that I want to send you. That SARS-CoV-2 infection model in humans, we found that there is a higher number of infected cells in the case of humans, and because this infection is like spreading across the tissue, will cause like a disease and sickness in the individual. But because bats can control the spread of the virus and only get like a single or a few cells infected with a high amount of virus, this will allow to persistence of the virus within the individual without producing any sign of disease because there are not so many cells infected. That's the main message that I want you to catch from this work. And finally, I would like to thank you all the people involved in this work. I will be glad to answer any questions.
Thank you very much, Iván. It's interesting theme. We have been in a pandemic situation, not so much time away.
Yeah, I hope nobody's asking me what's the origin of SARS-CoV-2. Yeah, hopefully.
You don't have the answer, of course.
Well, I can give you like what's like the main consensus, right?
Yes. Okay. We have still schools from Spain, Greece, and Croatia. What do you think about Ivan's research? Did you understand? Do you have some question, curiosity? They are clapping. You? Here. Okay. Go ahead.
So I have a question. Can bats make a resilience against other diseases like coronavirus?
Can you repeat the question, because there was like noise in the background and I could not listen properly.
Against other diseases.
Can you repeat again the question?
Can bats make a resilience against other viruses like coronavirus?
Yeah, I mean, actually, bats they know to resist like other viruses, like all these viruses that I mentioned that they are like reservoirs, such as Ebola and Nipah viruses. They are reservoirs, and these viruses they are not killing them, right? And they are not showing any sign of disease, but they are not resistant to all the viruses. There are like some viruses that they produce disease in bats, and actually they are killing them. Actually, there was like a virus found in, I think it was north of Spain. It was like, the Joviu virus. It's like an Ebola-like virus, and this virus killed all the bats in the cave, right? So it's like a very aggressive virus. Yeah, like the general consensus that bats can harbor like a lot of diseases. It's like partially true, right? Can harbor like some viruses, but not all of them.
Okay, thank you.